Hi, Stephen. Hi, Kyle.
Hi, John.
Hi, John, how you doing?
So welcome all to the Tribe Public webinar series event with Atossa Therapeutics. Today, we appreciate all of you that have already logged in and that will be logging in. And I'd like to introduce you to Dr. Stephen Quay, MD, Ph.D., founder, CEO of Atossa Therapeutics. That's going to be giving us a presentation today on Atossa and their work across a couple different fields, including COVID-19, as well as breast cancer. And we also have the CFO of Atossa Therapeutics, Kyle. Welcome, Kyle. And he's going to be helping out with a few of the slides in the presentation. And then we'll have a Q&A presentation, a Q&A session at the end of the presentation. Please feel free to send your questions through chat, through Zoom, or through email. I'll try to keep an eye on both of them as they come in. We'll address as many of them as we can. And we'll try to wrap this all up in about 30 minutes today, as promised. Stephen, if you wouldn't mind, let's go ahead and I'll share your screen with us,
Okay.
your PowerPoint. Can you pull that up and go right at it?
Let me do that for you, John. And let me get it full screen here. There you go.
Great. So why don't I start off with the first handful of slides and I'll turn it over to Dr. Quay. Again, this is Kyle Guzzi speaking. Thank you, John, for organizing this, and thank you, everyone, for attending. Slide 2 is a reminder that we'll be making forward-looking statements throughout the presentation. We're an SEC reporting company. I urge everyone to read our SEC documents. Slide 3. And by the way, our slide deck is available on our website under the Investor Relations tab. And so if anybody would like to look at the slide deck and follow along now or later, it's available there on Atossa Therapeutics website. So we're a clinical stage biopharmaceutical company focused on breast cancer and COVID-19. On the breast health side of the business, we recently announced positive phase 2 data. We have 2 phase 2 studies underway in breast cancer, and we're planning another phase 2 study for the second half of this year. On the COVID-19 side, we have 2 therapeutic programs underway, one called AT301, another called ATH201, and we have positive in vitro test results for both of those programs. Next slide. So summary of the leadership here, we recently added Dr. Frazier to head up clinical, regulatory, and CMC. Dr. Frazier has over 20 years of experience with leading biotechnology companies. I'm, again, the CFO and general counsel. I've spent most of my 30-year career as a lawyer counseling biotech companies on capital markets and M&A. And then Dr. Stephen Quay. Steve's the founder, CEO, and president of Atossa. He's a Ph.D. MD. He did his he did his residency at Mass General, which is Harvard's largest teaching hospital. At night, he did postdoc work at a lab at MIT under the direction of a Nobel Prize winner. He then went on to teach at Stanford for almost 10 years. And after that, he started a series of companies. He has invented and taken to market 5 FDA-approved drugs and 2 nasal spray products. And Steve will talk more about our nasal spray program in a minute, which will make you all wonder why you're listening to me rather than Steve. But I'll turn it over to Steve in just a couple more minutes here. Next slide. So corporate summary. We have no debt as of March 31st. We had about 9.4 million in cash at March 31st. Our fully diluted market cap's about 76 million as of yesterday's close. And we're based here in Seattle, Washington. Next slide. So a summary of our clinical pipeline for our leading programs. As you can see, we are in the clinic with our breast cancer programs about mid-stage of development, and we are very near entering the clinic with our COVID-19 programs. And with that, I'll turn it over to you, Steve.
Thanks very much, Kyle. I appreciate it. And John, I appreciate the opportunity to speak today. Our two programs in COVID and our two programs in breast health, breast cancer, are addressing very serious, big problems. ATH 201 for COVID ventilated patients is for the sickest patients, those on a ventilator or trying to keep people off a ventilator as they begin to lose their pulmonary function. And as we know, unfortunately, at this point in time, there's over 600,000 deaths from COVID at the present time. Our AT301 nasal spray works at the other end of the spectrum in newly diagnosed patients, or even as a preventative to try to prevent patients from getting ill. And we know that COVID is still a pandemic that's growing in many, many countries, including the US, with over 50 million current cases. Switching to breast health, we have an oral endoxifen program to lower mammographic breast density. Density has two issues. It interferes with the sensitivity of the 39 million mammograms each year, making it harder to find cancers. And we think an important adjuvant to mammography would be an oral dose of endoxifen in women that have very high density. And the other indication is that density itself is an independent risk factor of increased cancer. The dense environment does something biologically to enhance the presence of early cancers and carcinoma in situ. So we're developing programs for both of those conditions. And finally, there's what we call a window of opportunity, which occurs between the date of diagnosis and the treatment, which is 3 to 4 or 5 weeks after that diagnosis. Excuse me, when a woman can take an oral dose of endoxifen and actually start to kill cancers the day after she gets her diagnosis, so that by the time she gets to surgery, there's the cancer's perhaps potentially smaller, and it's certainly much less active. Before we get into our programs, I'd like to summarize what we've learned in the last 6 months, especially with COVID, the COVID disease itself. Thanks to the work of thousands of scientists literally all over the world, we now know that COVID has 3 stages basically, and I'm using my pointer. I hope you can see me circling stage 1 in the lower left of this slide. This is the initial infection, and it actually is a nasal infection. From the time of actual absorption, day 0 as they call it, to days 2 or 3, the infection is living and reproducing and growing in the nasal mucosa and the back of the throat. If you don't stop it at that point in time, and it's really hard to do so unless perhaps you have memory T cells from a prior COVID infection 20 years ago, you go to stage 2, where it then begins to move from the nasopharynx and nose down into the lungs, and if severe enough, can qualify for a pneumonia. This is a 3 to 10 day process, and again, by 10 days, a person with a strong immune system will have begun to fight this, and the mild to moderate cases that recover without going to the hospital are stopped at this stage. They're usually not stopped at stage 1, but they're stopped at early stage 2. Now, what happens here, if your immune system still hasn't caught up, or if you have pre-existing conditions that may make it harder for you to clear this out of your lung, mechanically even, you then go into the systemic phase, which is day 10 through, we're seeing patients now coming out of the hospital after 90 to 100 days in the hospital. And then this is also, of course, a stage where the mortality exists. So if you think of the disease in these 3 stages, it sets you up to think about 4 areas of therapeutic need, which Atossa is trying to address in a very systematic fashion. So the first is a nasal spray to take before the diagnosis or at the time of diagnosis. This is to treat very early disease, to prevent it from going into the lungs, and maybe even as a prophylactic in people, for example, medical personnel, people at the airport that are in contact, service-facing people, to actually prevent disease. If you end up at the hospital, you then need systemic treatment to prevent that systemic disease, to keep it in the lungs, so to speak. Now, you also probably need inhalation therapy, because as the disease develops in your lungs, you begin to lose the ability to oxygenate your blood. You need either additional oxygen, you need a nebulizer, or eventually a ventilator. We have a program in this area which we think is very important for this phase of the operation. And frankly, there's a new phase that's beginning to appear that we're paying some attention to, and that is the post-recovery phase. Even patients with mild to moderate COVID-19 never go to the hospital at up to 90 days after their initial infection, have residual cardiovascular disease. And we think that as the 15 million gets even larger and as more people recover, this is going to be an important medical need that currently is largely being ignored. So this is how Atossa Therapeutics sees this disease. And of course, there's a vaccine that would go up here somewhere, and we don't have a vaccine program at this point in time. So let's go through the programs. ATH-201 is for COVID patients on ventilators to improve lung function. So that's operating down in this box down here. We've successfully completed in vitro tests to show that this stops the virus from getting into vero cells in the laboratory. That doesn't prove that it will be safe or effective in humans, but it is the screening test that every drug has to get through before it gets into preclinical and clinical studies. This is a true two-drug combination of already approved drugs given by other routes or for other indications. So our development path is assisted by this prior FDA experience, but it also requires the fact that because it's a two-drug combination, we may often have to test the drug separately and then the combination itself. We are under contract in New York Hospital for a clinical study and have applied to the FDA to open that initial study with one of the key components, which is this process is currently pending. And we're working diligently with the FDA, and of course they're very responsive to the acute, immense medical need of COVID-19. We do have provisional patent applications surrounding this. So this is the data. This is what it looks like. I think did I move everybody down so you can see this? So this is AH-201 keeping the virus from getting into vero cells and replicating. When you don't have either of these around, what you see is the green, which indicates a fluorescent protein. One of the M proteins is made includes a gene that makes it green under these conditions. So you see that this is a very potent inhibitor, and that as you dilute it over and over again, you finally do get virus growing, which is a good positive test for an experiment like this. But this is in vitro in this same setting, 4 times more potent than remdesivir, which is of course the current IV drug. Switching back now to A2301, our COVID-19 nasal spray. This targets infections in the nasal mucosa. It literally is a chemical nasal mucosa vaccine, as we like to think of it. It's used for outpatient and home use. We're expecting that it will be used 3 times a day for a newly infected patient. So, every 8 hours they would take a spray. It has shown successful in vitro work. And we are working with a group called CRO Avance in Australia for a clinical trial to be planned in the second half of this year. We also have patent applications around this, and Summit Biosystems in Kentucky is our contract manufacturer of the clinical trial supplies. People often ask, how do you compare a vaccine to a therapeutic? So I made this chart here, which uses one of the many vaccines, the mRNA 1273 from Moderna, in our 2 programs. So as indicated in this table, the vaccine, the mRNA 1273 is of course a vaccine. Ours are antiviral therapeutics. The primary target for all 3 of these drugs is the spike protein. Excuse me. So the intent of a vaccine is to have you make antibodies and T cells against the spike protein, because that's the antigen that's being immunized. And our formulations contain chemicals that modify the binding sites, the receptor binding domains of the spike protein in a very similar fashion. Now, in terms of secondary targets, the current vaccines don't have any. So they're relying all of their efficacy on this one target of the viral envelope protein, the virus envelope particle. We are also directing chemicals that are inhibitors of the proteases that are host entry gene products. So the virus, as you know, is inert. It's not really a living thing, but what it does is it is designed to trigger host enzymes, proteases, little scissors that clip the spike protein and release a tunnel that allows the RNA to get into the cell and infect the cell. So our therapeutic products have that double approach of not only inhibiting the spike protein directly, but preventing the host enzymes, hopefully, from doing their action. The technology here is a messenger RNA. So what this product is doing is teaching cells in the muscle to make something that looks like the spike protein and contains the protein component of it in a fashion that incites an immune response. Ours are simply chemicals that block binding sites in the receptor domain or block enzymatic activity on the host surface. There are no previous FDA-approved drugs using messenger RNA, which doesn't mean that there won't be a first, just like there was a first insulin from E. coli. But it is a new technology for all of us in the medical community. All three of the products stop infections in vitro. We don't know about the durability of this. One of the things that I'm watching for very carefully is the neutralizing antibody titer immediately after vaccination and what it looks like a month or 2 or 3 months later. There's something very diabolical about COVID-19, both in recovered patients and in the vaccine studies that I've seen, where the neutralizing antibodies seem to tend to diminish. And whether that'll have an impact on infectivity, we don't know, but it's something that everyone should be watching in thinking about the efficacy of a vaccine. The other thing is, of course, mucosal activity. As we now know, this virus spends 3 days in the nasal mucosa, where the primary antibody is IgA, not IgG or IgM. And so when the companies and folks are reporting on these vaccines, I'm always looking for trying to find out what the IgA titer is, which is usually not reported, because that's the one that will be indicating a mucosal activity. Ours, of course, is expected to be effective when used. Basically, I'm using leveraging the experience I had with the other nasal spray products that are FDA approved to bring to bear on this therapeutic. Now let's switch to breast cancer because it still is an ongoing problem. 1 in 8 women experience breast cancer in their lifetime. There's 268,000 women in the US diagnosed. It's the second leading cause of death for women in the US. Our phase 2 WOO, or Window of Opportunity study, is involved in taking advantage of the time between diagnosis and surgery. So during this time, endoxifen is given at 4 milligrams orally on a daily basis, and the primary endpoint is to reduce the growth activity of this cell population. It's called Ki-67 is the measurement in the laboratory, and we obviously do secondary endpoints of safety and durability. In the interim results that we reported earlier, there was a 74% overall reduction in Ki-67 activity, which was highly significant even in only 6 patients. So this is extremely encouraging. All of the patients achieved over a 50% reduction, and very critically, they all had less than 25% activity at the time of surgery. So when the tumor was removed, less than 1 in 4 cells was still dividing, was still giving a Ki-67 signal. In a meta-analysis of literally 70,000 breast cancer patients, this number is the magic number for predicting that you will not have a recurrence. And in our little study of 6 patients, everyone achieved this number of less than 25, which is highly clinically significant because of the tie-in between this activity at the time of biopsy and a recurrence 5 years later. There was an average of 22 days of treatment, which is relatively short treatment, about 3 weeks between diagnosis and surgery. The range was 16 to 40 days. And again, there were no safety signals. The tolerance of endoxifen has been extremely good, and in fact, does not, in our clinical trial experience, have the vasomotor or hot flashes, night sweat side effects that are typically seen with SERMs like tamoxifen and other drugs. The other study we have ongoing, we reported today in a press release, is an expanded access patient where the FDA gave us permission to give this woman 51-year-old premenopausal woman with an ER-positive breast cancer who has the genetic predisposition in her liver called CYP2D6 star 4 star 4. Not a very clear name, but what this means to the doctors, the oncologists, is her liver cannot metabolize the most common breast cancer drug given in a proper fashion to make it active. So she is not a candidate for that drug. Her physicians came to us. We gave this product to her again in the neoadjuvant setting before surgery. So for 20 days was her window of opportunity. Therapeutic levels were achieved within 6 days. So it really gets acting quickly. And again, her Ki-67 was below 50%. It was below 25% threshold. Again, it was very well tolerated. Her physicians asked if we could continue her on this drug because her tumor was so responsive to this treatment. And so we did. We got FDA permission under an expanded access IND to give her 4 milligrams of endoxifen for a full 18 months now. And at this point in time, she's had no recurrences based on clinical exam or mammography. She's had no signals of safety or toxicity based on laboratory tests that we've been doing along the way. And again, it's been very well tolerated with literally no vasomotor symptoms. The other important thing that endoxifen does is to prevent this problem. Mammograms need help. Finding this cancer in a breast that's this dense is literally impossible. And most cancers that are missed, in fact, are in high dense breasts. Endoxifen lowers density, and we believe in doing so will help us on mass cancers to make cancers earlier to find in these women. The other positive effect of lowering density is what's shown here. So this is a low density breast, a high density breast. This is set at 1.0, so we can compare cancer incidence as you go up in density. And these are the results. It goes from a little over 1 to almost 5-fold increase in breast cancer simply by having a dense breast. So we believe by being able to give oral endoxifen for 3 months, 6 months, we don't know the duration, that's what clinical trials are all about, but by having that opportunity, we can lower the density and presumably lower the future risk of breast cancer. Kyle, why don't you go over the milestones and updates, if you would, at this point?
Thank you, Steve. Yeah, you'll see that we essentially have 6 milestones coming up during the remainder of 2020. For ATH-201, we expect to receive regulatory approval to open the study and then start a study in the 3rd quarter. For our COVID-19 program, AT301, again, in the 2nd half of this year, we expect to receive regulatory approval to open a study and then actually open the study. And the same with a breast health program for breast density. In the 2nd half of this year, we expect to receive regulatory approval and then open the study. So over the course of the next 5 months, we have 6 very significant milestones that we're working towards. And, looking at our time, I wonder if we should turn this back over to John to see if we can tee up some questions.
Yes, gentlemen, thank you so much for the presentation today. I think you probably addressed a lot of the questions that people have, but let me relay a couple of these. Here's the first one. By the way, if you guys didn't realize this, we have people literally have come in from around the world, from Brussels, from London, Singapore, across the US. This is such a great format that I'm excited to bring this to the tribe and to Atossa shareholders, et cetera, to learn more. But it's very exciting to see all the different opinions and everybody coming in and around the world to sort of tackle these issues that you are working so hard to deliver on treatment. So, kudos to you guys, but also it's thank you all that have joined today. One of the questions here is, gentleman has pushed forward, it says, in addition to dealing with, since competitors who are manufacturing drugs like hydrochloroquine and remdesivir, can your mixed combination of the already FDA approved heparin, et cetera, be used to go to treatment for most hospital admissions is the thought, or that are COVID positive, or is it only for those rare cases? How would you address that question?
Yeah, so the ATH-201 formulation is specifically designed to block entry into pulmonary cells and or to improve lung function. So these are patients who are in the hospital, seriously ill, sufficiently ill with the condition to either require a nebulized oxygen to assist or a ventilator. So our endpoint is simple things like keeping people off of the ventilator or getting them off early. As we know, there's some variability about the death rate on ventilators, but the variability is the lowest I've ever seen is 10 to 15%, and the highest I've seen is 30 to 40%. So that's a very high medical need that we think our product will address in a very fine fashion. So we're anxious to get that study started.
Okay, thank you, Stephen. One of the other questions here is if the development of AT301, ATH201, and endoxifen oral are successful, when do you, I guess, can you sum up where you are with the FDA and when you might think that is reality for gaining the approval of the long-term process? Can you sort of speak to that process in your experience?
Well, it's very hard because there's never been anyone who's developed drugs during a COVID pandemic that I'm aware of. And the FDA didn't exist in 1918 when they had the Spanish flu, so we don't have any precedent from that. What we need to do is get clinical data that's impactful as quickly as possible. And once we have that in our hands, that becomes the leverage that we have to see what is the next step to get approval, what do we need to do, but we can't speculate on what the timeframes will be without having the clinical data in our hands.
Okay. Switching on the financial side of it, one of the questions has come in to fund these programs. How are you currently set up as a company from the balance sheet? And I know you'll be reporting again here in August, but can you speak to what is in the public domain regarding your funding of these programs? Maybe that's a question for Kyle. Or
yeah, I'll tackle that one. I mean, you'll see that historically, based on our SEC filings, we've burned $700,000 to $800,000 per month in cash. So we're a small, lean company that's very cash conscious. We will file our next Q in about 2 weeks, and so you'll see the expenses and the cash burn rate from this last quarter. We're an SEC compliant, NASDAQ listed company. The capital markets are extremely strong, I guess is what I would say, based on the advice we're hearing from investment bankers. So we will have access to capital when we need it, I guess is what I would say. Our cash balance in relation to our burn rate is actually fairly healthy for a small biotech company in mid-stage development. So our cash balance is pretty strong now in relation to our burn rate, and we'll raise money when we need to.
Okay, thank you, Kyle. Switching back to the science portion of this, Stephen, one of the questions is, can you speak a little bit more about your the nasal approach as being a preventative? Again, can you speak just reconfirm what maybe what you'd said about that and what your approach is here, and then also maybe speak to your partner that you picked, Summit, to help you push that process through.
Sure. So the key design features for a nasal spray therapeutic in this particular space includes residence time. So what we want to do is design it with polymers that allow it to stay on the nasal mucosa 2, 3, 4, maybe even 6 or 8 hours. So, excuse me, that's one of the sort of the fundamental baseline principles. Because absent surfactants and other kinds of excipients that you put in these, the nasal mucosa is cleared about every 30 to 40 minutes by natural ciliary action. And I've written review papers for other people who want to design drugs in nasal formulations. So that's just the foundation. what we've done is we've identified some key targets on the receptor binding domain of the spike protein and three key proteases, ACE2, furin, and TMPRSS2. I couldn't come up with a better name than that, but these three host proteins on the surface are each proteases, so they're like scissors that cut the spike protein and other proteins of the SARS-CoV-2, and they allow the virus to get into the cell. So by preventing these proteases from acting on the spike protein and other proteins of the viral particle, and by blocking the viral particle itself, like a mucosal antibody would do, we believe this twofold combination provides a very high safety margin that the virus simply gets caught up and doesn't infect the cell. And remember, these are literally like Pandora's box, which sat, in the history book, sat inert for hundreds of years till someone, opened the lid, and then the world was fighting the rest of the movie, trying to figure out if it would survive. These viruses are completely inert if you don't clip their activating pieces on the surface, and we've got inhibitors that block all three of the major activators.
Thank you, Stephen. One thing, I know we're getting tight on time here. Look like we're about a minute over, but one of the things I wanted to make sure that everybody recognized that you've recently published the COVID survival manual.
Yes.
And there it is. And
I actually have my own copy. It's
very good. It's a great read, guys. And if all of you haven't ordered it yet, after this, for participating in this webinar today, Dr. Quay has offered, he's going to send out a discount code. I'll get it out to you via email and give you the links to it if you'd like to order that. That will give you a significant discount. Some of the proceeds are going to charities as well. So I encourage you.
I'm sharing it with Team Rubicon, which is a group of veterans helping in communities of need in COVID and other national emergencies. So it's a good group.
Thank you. Also, I'd like to remind everyone that this is an ongoing process. So the tribe is about the companies you care about. And so through the process at our website, we have the wish list process. If you'd like to hear from Stephen again sometime in the future, and Kyle and Atossa, please submit your interest in doing so. I would anticipate that they might actually jump back on here after accomplishing a couple of these milestones in a fairly quick fashion, and hopefully, and then help us gain better understanding as to where the direction is going, and it's a continued process. I appreciate everyone that's participated today. I appreciate you, Dr. Stephen Quay and Kyle, to taking the time to share this and for your work at Atossa. I'm a shareholder too, everyone. And so, I'm very interested in seeing this company be successful, and most importantly, that they'll be able to bring these treatments through the FDA process and ultimately to all of us. So again, thank you all for participating. Thank you all for being part of this this process. I would leave Stephen, if there's anything you would like to sum up, maybe sum it up right now. And again, I bid everybody adieu, and please send me emails, questions, etc. after this if they didn't get addressed, and we'll do our best to get those addressed.
Yeah, John, I mean, I just want to be sure I emphasize here that Kyle and I are maybe are the face of Atossa, but the people at Atossa are the ones that get things done. And so I have the best team of folks that I've worked with in my entire career. It's really it's a blessing to go to work. We go to work virtually these days, but to go to work every day and work with the dedicated people at Atossa Therapeutics. So thank you, John. Thank you, Kyle. Thank you, folks, for your questions and your support of Atossa. We look forward to speaking to you again soon.
All right. Thanks. Thank you all. I'll see you soon.
Thanks, John.